Hi everyone, today we'll be talking about virus vectors, in particularly the vectors derived from retrovirus HIV, adenovirus AD5, parvovirus, adeno-associated virus, flavivirus, West Nile strain Kudjun. They all have different features because they are derived from different viruses, and these different features means that they are suitable for different roles in biomedical research and public health. So for the retroviruses, they have the ability to integrate into the host genome, which is not desirable if you're infecting mammalian cells because you want control to some degree. So essentially, the regeneration of replication competent retrovirus is an issue. So you would delete some of those GAG, PO, and OMV proteins in its genome and then replace it with heterologous genes. And then you would transfect it into cells expressing GAG, PO, and OMV proteins. Essentially what this does is that it limits the generation of replication competent retroviruses. And then the resulting cell line would then produce these virus particle-like molecules, which can then be harvested and concentrated and used to infect cells. So they have been shown to play a good role in gene correction therapy because they don't have like a strong cytotoxicity or like vector-induced immune response. They have been used for cancer therapy because it can be produced to fairly high levels quickly to kill those tumor cells and induce cytotoxicity on them. So it actually has very select cytotoxicity as well as pathogenicity. And it is also used for vaccine development because it has high immunogenicity when used for this purpose. But essentially you get the turning off of replication genes by deletion of integration genes, so that you don't get the interference with host DNA by deleting the 5' LTR region U3 and the 3' LTR region partially. And it has 4 different plasmids, and the role of expressing packaging proteins in separate plasmids also helps to prevent the regeneration of replication competent retrovirus and endogenous retroviruses. And they have also been shown to have limited envelope tropism, which is undesirable. So what we have found a way to bypass that is to code for the pseudotype vector particles with pantropic envelope VSV alpha protein. And what else? And also we inactivate some of its TAT proteins, which activate its transcription, and instead use host self-activating factors to control. Adenovirus, it has many advantages and desirable features, including the fact that it can be produced to very high titers, the fact that it has a large insert sequence, and also it has high immunogenicity, which makes it good for stimulating a strong humoral and T-cell response in vaccine therapy and cancer therapy. And it also, can efficiently transduce, which is awesome, and can affect non-dividing cells just like retrovirus HIV. But then the disadvantages is also its high immunogenicity. So it can't be used for gene therapy because that might kill people. And also the fact that over 40% of us have been infected with ADV5 in the past. So we might already have a human response against it. So it might not elicit the desired features. So adeno-associated virus, it's capable of integrating into the chromosome and persistent infection, which makes it good for gene therapy, but again, it's a bit worrying in terms of biosafety. But it doesn't demonstrate any insertional mutagenesis, and it is fairly safe and also infects non-dividing cells. But disadvantages include, possible death. There was one case of possible death recorded from adeno-associated virus vector therapy. It has a very small insert sequence. It has a lower titer, and it also is very labor intensive. Finally, for the remaining RNA viruses, which don't include the retrovirus, because that shows an integration step, these are very good because they replicate in the cytoplasm, and being in the cytoplasm, you don't have to worry about insertional mutagenesis of host DNA, etc. And plus, they can replicate very quickly given their small size, producing virus-like particles, naked DNA or RNA, which can be used to infect cells. And the example we looked at is the West Nile virus, and this one is good because it has been shown to be used for some vaccines and cancer therapy. Yeah, that's it. And then, so to sum up, you have vectors derived from viruses, and these vectors have been engineered to elicit a specific response that makes it efficient for gene therapy or cancer therapy or vaccine therapy. And the basic stages include removing the bits that you don't want, and these generally include genes that increase the possibility for pathogenicity and replication competent viruses. Then you would keep all those genes that you need, such as the packaging, such as flanking all those necessary structural genes if you do decide to keep them with cis-acting genes and packaging signals, while still keep making sure that you stay within the insert limit. You might choose to add a promoter which encourages its production. Then you would produce this within a cell line that expresses enzymes that bind to promoters and facilitate expression. This expression can be temporary or permanent, but basically you get some virus-like particles being produced, which you can purify and concentrate using cesium chloride. And there are definitely issues, but we have figured out some ways to bypass these limitations. Thanks for listening.